How?
Peptide-functionalized liposomes targeting breast cancer metastasis. Hey everyone! Welcome back to our channel. Today, we're diving into some groundbreaking research on breast cancer metastasis and how new peptide-functionalized liposomes could change the game in cancer treatment. So, grab a snack, get comfy, and let's get started. Breast cancer is a major health challenge, especially when it comes to metastasis, which is when cancer spreads from the original site to other parts of the body. Researchers are constantly looking for innovative ways to tackle this issue, and today, we're exploring a fascinating study that utilizes peptide-functionalized liposomes. First off, let's break down what liposomes are. Think of them as tiny bubbles made from lipids, which are fats. They can encapsulate drugs and deliver them directly to target cells. This targeted delivery is crucial because it minimizes side effects and maximizes the treatment's effectiveness. Now, what's the deal with peptides? In this study, researchers used a specific peptide called DV1 that binds to a receptor known as CXCR4. This receptor plays a significant role in cancer cell migration and metastasis. By attaching the DV1 peptide to liposomes, they created a new type of treatment that could potentially inhibit the spread of cancer cells. So, how did they do it? The researchers started by creating liposomes that had a special component called DBCO. This allows for a click reaction with the DV1 peptide, effectively attaching it to the liposome. They then tested these liposomes on two aggressive breast cancer cell lines, MDAMB231 and MDAMB436, as well as a non-cancerous cell line, MCF10A. After treating the cancer cells with these DV1 functionalized liposomes, they monitored how well they bound to the cells and how they affected cell behavior. They used techniques like dynamic light scattering to measure the size and integrity of the liposomes and flow cytometry to analyze cell binding. But that's not all. They also conducted in vivo studies using mice. They injected the mice with cancer cells and then treated them with the DV1 liposomes. The results were promising. The mice treated with the DV1 functionalized liposomes showed significantly reduced metastasis compared to control groups. So, what's happening at a molecular level? The study found that the density of the DV1 peptide on the liposomes was crucial. Higher densities led to a stronger inhibition of key proteins involved in cell migration, such as P115-ROGF and P85-PI3K. This means that the way these peptides are organized on the liposomes could dictate how effectively they can block cancer cell movement. Now let's specifically discuss the synthesis and characterization of DV1 functionalized liposomes, or LDV1 for short. So, what exactly are liposomes? Think of them as tiny bubbles made up of lipids. These are the same fats that make up our cell membranes. They can be used to deliver drugs or other therapeutic agents directly to specific cells in the body. In our case, we're focusing on a special type of liposome that's been modified with a peptide called DV1. Now, let's get into the nitty-gritty of how these DV1 functionalized liposomes are made. The process starts with a compound called DSPE-PEG-2000NH2. This is a lipid that has a polyethylene glycol chain attached to it. Why PEG, you ask? Well, PEG helps improve the stability and circulation time of the liposomes in the bloodstream. Next, we combine this DSPE-PEG-2000 with another compound known as dibenzocycleuctine, or DBCO for short. This is where the magic of click chemistry comes into play. Click chemistry is a super efficient way to link molecules together, and it's perfect for our purpose. Once we have our DBCO lipid ready, we create what's called LDBCO liposomes by mixing this with another lipid called DOPC. This mixture is then processed using a method called extrusion, which essentially squeezes the lipids through a membrane to form uniform liposomes. Now, here's where we get to the fun part, attaching the DV1 peptide to our liposomes. We use the azide form of the DV1 peptide, which reacts with our DBCO lipid in a click reaction. This step is crucial because it allows us to attach the DV1 peptide to the surface of the liposome in a specific orientation. After the reaction, we need to confirm that the DV1 peptide has been successfully attached. To do this, we use a technique called dynamic light scattering, or DLS. DLS helps us measure the size and stability of our liposomes. But that's not all. We also want to visualize our liposomes. For this, we can use electron microscopy techniques like scanning electron microscopy, SEM, and transmission electron microscopy, TM. These methods give us detailed images of the liposomes, helping us confirm their size and shape. Now, let's talk about the actual binding and uptake of these DV1 functionalized liposomes. We tested how well these liposomes bind to two types of breast cancer cells, MDAMB231 and MDAMB436. It turns out that the density of the DV1 peptides on the liposome surface is key. We found that at a density of 24,000 DV1 molecules per square micrometer, or LDV1-24K, we achieved the highest binding affinity to these cancer cells. This means that the liposomes are very effective at targeting the cancer cells, which is exactly what we want for potential therapies. So, in summary, we synthesized LDV1 liposomes by combining various lipids and using click chemistry to attach the DV1 peptide. We characterized these liposomes to ensure they're the right size and shape, and we tested their ability to bind to cancer cells. Now let's discuss DV1N3 and its functionalized liposomes, known as LDV1. These compounds are making waves in the field of cancer research, particularly for their role in regulating cell migration in breast cancer. First off, let's talk about what DV1N3 is. DV1N3 is a peptide that has shown a strong ability to bind to a specific receptor on cancer cells called CXCR4. Now, you might be wondering, what is CXCR4? Great question. CXCR4 is a type of receptor that plays a crucial role in cell signaling, particularly in processes like cell migration and metastasis, which is when cancer cells spread from the original tumor to other parts of the body. In the study, researchers found that DV1N3 not only binds to CXCR4 but does so with a higher affinity than some existing treatments. This means it could potentially be a more effective option for targeting breast cancer cells. Now, how does DV1N3 affect cell migration? Well, migration is a vital step in the metastasis process. It involves cancer cells detaching from the original tumor, moving through tissues, and eventually establishing new growths in other organs. The researchers conducted experiments to see how DV1N3 influences this migration. They compared DV1N3 to a control, which was a scrambled version of the peptide that doesn't have the same binding ability. What they found was that at an equal concentration, DV1N3 inhibited migration by about 55%, while the control only managed to inhibit it by 27%. This indicates that DV1N3 is quite effective at preventing cancer cells from moving and spreading. But that's not all. The researchers also created LDV1, which are liposomes, tiny spherical structures made of lipids that can carry drugs or peptides. By functionalizing these liposomes with DV1N3, they aim to enhance the delivery of the peptide to cancer cells. They tested different densities of DV1 on the liposomes to find the optimal binding capacity. They discovered that a density of 24,000 molecules per square micron, referred to as LDV1-24K, exhibited the highest binding to breast cancer cells. This specific density allowed for stronger interactions, leading to an 84% reduction in cell migration compared to controls. Now, let's talk about the implications of these findings. By using LDV1-24K, the researchers observed a significant decrease in breast cancer cell migration in vitro, which means in a controlled lab environment. This is a promising step towards developing new therapies that can prevent metastasis without the need for traditional chemotherapy, which often comes with harsh side effects. Additionally, they noted that LDV1-24K did not show any toxicity to normal cells, which is a crucial factor in cancer treatment. This specificity means that it could potentially minimize damage to healthy tissues while effectively targeting cancer cells. To sum it all up, DV1N3 and its functionalized liposomes, LDV1, represent a novel approach to tackling breast cancer metastasis. By effectively inhibiting cell migration through targeted binding to the CXCR4 receptor, this research opens up exciting possibilities for future cancer therapies. Thanks for tuning in today. If you found this information helpful, don't forget to like, subscribe, and hit that notification bell for more updates on groundbreaking research in science and medicine. Until next time, stay curious.
PharmaPal.